everyone, let's talk a little bit about proteins. So proteins are long chains of amino acids, and amino acids are considered building blocks for proteins. So amino acids, we're going to actually talk about the different amino acids. There are 20 in total that are in the human body that are needed for continued functioning. 9 of those 20 are essential in the diet, meaning that they have to be consumed and the body cannot synthesize them. But the other 11 can be synthesized by the body. And so some amino acids can be broken down and rearranged a little bit so that other amino acids can be built, but some amino acids have to be ingested. That's the only way to get them. So there are 5 components to an amino acid. Number 1, there's a central carbon atom. So we know that carbon can make 4 bonds. So carbon is going to be in the middle and carbon will attach to 4 different things. One of those things is going to be a hydrogen atom. One of them is going to be what we call an amino group. Amino group is a nitrogen with 2 hydrogens attached. So the carbon will attach to the nitrogen and the nitrogen will attach to 2 other hydrogens. And the nitrogen actually, with that number of bonds, is considered full. So it has 5 valence electrons and then it has 3 empty spaces, so it can make 3 bonds. We also will attach a carboxylic acid group to the carbon, the central carbon atom. We see a carbon atom that's attached to an oxygen with a double bond and then attached to another oxygen which is attached to a hydrogen. And then lastly, we have what's called a variable group or an R group. So the R is representative of kind of an unknown. So in the 20 amino acids, the first 4 pieces, the central carbon atom, the hydrogen atom, the amino group, and the carboxylic acid group are all going to be exactly the same. But the thing that differentiates the 20 amino acids is what the R group represents. So the R could be a hydrogen. The R could be a methyl group, a carbon attached to 3 hydrogens. So for each of the 20, it's going to be something different. So we're not going to draw them all, but it's just useful to sort of understand what we're talking about when we're referring to amino acids. So amino acids are small and they're water soluble. That's another important thing to keep in mind as we think about the future of how we're going to study the human body. Now, if we were to attach 2 amino acids together, we'll connect them by way of something called a peptide bond. So if we have 2 amino acids that are strung together, we call it a dipeptide. If we have 3, we call it a tripeptide. And if we have 3 or more amino acids strung together, we can call it a polypeptide. So technically speaking, a tripeptide can be also called a polypeptide. And if there's more than about 100, we'll call it a protein. So really, the distinction for protein is just to show that there's really a bunch of amino acids that are strung together, and it could be many, many more than even just 100. So what's interesting about proteins is that we string these amino acids together. You can see how the proteins can get really big really fast because, there's 2 carbons for every amino acid. So already we're looking at 200 carbons, and it's just kind of a crazy thing, like how quickly we can just string these atoms together and then make a big molecule. So amino acids are small, but proteins can get pretty big in relative terms. Okay, so once we string the amino acids together, what we get is a linear chain, and we call that the primary structure. So the primary structure is just a sequence of amino acids in a linear chain. And we'll just draw them as little boxes connected to each other, but we'll remember them as the carbons and the nitrogen and the hydrogen and stuff that we drew in the previous screen. So a protein is not useful in its linear chain. So we actually need to fold it up into something that's a little bit more interesting. So the secondary structure is sort of like we get some bonds between different amino acids in the chain, and it's our first piece of the folding process. And there are 2 general types of secondary structures. There's the alpha helix and the beta pleated sheet. And so the alpha helix is just sort of like a cylindrical or circular pattern for how the amino acid chain will fold. And the beta pleated sheet is more of like a zigzag type of shape, a little bit more boxy looking. From there, we'll actually develop our tertiary structure, And the tertiary structure is a complex coiling and folding process that gives us a three-dimensional shape. So the tertiary structure for some proteins is kind of enough. That's where it will stop. But for other proteins, it will actually have multiple subunits. So we could take multiple primary structures of amino acid chains and fold them up, and then in a quaternary structure we can link them together. So a quaternary structure is like a couple of different tertiary structures of proteins linked together to make a bigger protein. So a good example would be hemoglobin. So hemoglobin is the molecule that is responsible for carrying oxygen in red blood cells, and it's actually made of 4 subunits, 2 alpha subunits and 2 beta subunits. And so think of that as 4 tertiary structures stuck together to make this large protein called hemoglobin. So the folding of proteins, giving it an interesting shape. That shape is what's going to determine the function and the properties of that particular protein. So the protein becomes functional, becomes usable, is able to do a job once it's all folded up and in its three-dimensional shape, or possibly with multiple subunits. Denaturation is a term that refers to the unfolding of protein. So small changes in ionic composition, temperature, pH. If we suddenly have a drop in pH and we have acidosis, we can cause the proteins to unfold and become non-functional. So denaturation refers to the unfolding of proteins in the presence of these types of environmental changes. And denaturation can be critical because sometimes if the protein is exposed to these conditions for a prolonged period of time, the protein will unfold and not be able to fold back up again. So we lose the functioning of our proteins. And we're going to show in the next sort of couple of lectures the incredible significance of proteins in the human body. They make up so many of the structures that do these little cellular functions, and if we don't have them, or they start to break down because of a change in the environmental conditions, the body doesn't have much room to sort of compensate for that and keep up with it. All right, so that wraps up our discussion on proteins. Next up is nucleic acids and high energy bonds, and then we'll start building some cell structures. So I hope you enjoyed it. Thanks for watching.